So, welcome back for part two of the macromolecules lecture. Today I'm going to talk a little bit more in depth about the structure of the different macromolecules. Again, I'm not going to go into too far into details because this is all stuff you should have learned already in Biology 1. But we will be looking at the different pieces that make up the different macromolecules and how those pieces play into the function of each macromolecule. So the first thing that we need to address is the general structure of all the macromolecules. And each of these macromolecules are what are called polymers. Many mers, pieces maybe, right? So many pieces, many parts. Okay. The monomers are these smaller units that form the building blocks, the bases of these polymers. So I got these monomer units that clump together into larger pieces to form the polymer. So for instance, carbohydrates are a polymer. And there's a couple different forms of carbohydrates, right? There's sugars, there's starches, and then there are structural polysaccharides. Okay. The monomer of carbohydrates are monosaccharides. Right? So the polymer is a polysaccharide, the monomer is a monosaccharide. For lipids, the monomer is a fatty acid. For nucleic acids, the monomer is a nitrogenous base. And for proteins, the monomer are amino acids. So today, we're going to look at the structures of these monomers and how they come together to create the larger polymer that is the macromolecules. So, we are back and we have talked about monomers and polymers. And so I want to take my monomers, my smaller units of these macromolecules, and shove them together into these long chains to form my polymers. And the way that this occurs in the macromolecules is through a very similar reaction for all of them, and that reaction is called a dehydration synthesis. Okay. And the way that it works is indicated within its name, dehydration. Right? We are removing a water. Okay. And so OH groups are very prevalent amongst these macromolecules. And the way that this works is that two OH groups from the monomers will be joined together via an enzymatic process, right? So this doesn't happen spontaneously. I'm creating a larger molecule, and when I create a larger molecule, I am decreasing the amount of entropy in that system. And so anytime you have a decrease in entropy, you have to couple it with a change in energy, right? And so a decrease in entropy uses up free energy. And the way that our body copes with this is by utilizing enzymes to catalyze this reaction and make it happen. And so this won't happen naturally. Living things make this happen through enzymatic reactions. And so when this OH group meets this OH group, they come together with the help of the enzyme. We pop off an OH from one side and H from the other, right? And that makes H2O, aka water, right? And then that remaining O is bonded to the other monomer. So I get this linked one monomer, two monomers. I now have a very small polymer. In this particular depiction, these are sugar rings, right? So these are monosaccharides. I link them through dehydration synthesis And I now have a disaccharide. Di meaning two, mono meaning one. So a disaccharide is the first step to building a polysaccharide. And so this dehydration synthesis is used throughout all of our macromolecules to create our polymer strands. Cool? So now we know how we build up the polymers through the process of dehydration synthesis. The next question is, how do we break these polymers down back into their monomers? And now, why would we want to do such a thing? Right? We just spent this time building them up. But when we do things like eat, right? So maybe I just ate a big old steak. Right? I want to break down those proteins from that steak and turn them into monomers that I can utilize. Right? And so I can use those resources for my own. I can build my own proteins using those monomers or the smaller components of those monomers. Right? And so as a living system, right, we need to constantly replace the particles and the substances that our body is using to build stuff. And we need to get that from the outside world. Right? And so we do that through things like breathing and eating. And so we need to take the polymers of other living things, break them down to their raw materials so that we can then build up our own polymers. Right? So the way that we take a polymer and break it back down into monomers is through a process called hydrolysis. Okay. And now if you break down the word hydrolysis into its components, the prefix hydro referring to water, and the suffix lysis means to split. Right? So whenever we talk about lysing, we're talking about splitting things. It's a suffix that we'll see a few times throughout this course. So I have splitting by water. Okay? And the way that this works is effectively by doing the opposite of the dehydration synthesis. I take my polymer, I shove some water on it, and that water breaks these bonds here and joins with this oxygen for him, and that leaves this guy. So the hydrogen from water has joined over here, and now I go back to my original monomer. And then the remaining OH from the water attaches to this guy, and I get this. And so now I'm back to my old starting point with these two monomers here. Right? And now this reaction increases the entropy of this system. I took something that was more organized and I made it something that was less organized. Okay? And so this reaction will actually happen spontaneously. Right? And so if I leave a carbohydrate out on a table in some water, eventually that carbohydrate is going to break down into its monomers. The rate at which this occurs naturally is not sufficient for living things, so we also promote this reaction using enzymes to speed it up, but this is naturally a spontaneous reaction. And so this is how we take a big polymer and break it back down into little pieces so that we can use those resources to build our own stuff. So now we're going to take a peek at some of the individual properties of each of the macromolecules. And so again, I'm not going into every little detail about these things. I'm not going to recover the basics. Actually, I have a video posted on this channel for my Biology 1 class that does review some of the more basic things about these structures. If you're interested, you can go check that out. But I'm going to start with carbohydrates right now. Okay. And so again, the monomer of carbohydrates are monosaccharides, right? Effectively these sugar rings here. And I can join these sugar rings together to make long, long chains of polymers, right? So polysaccharides. Now, carbohydrates in general have two different functions, right? I have carbohydrates that work as energy storage. Right? So I eat some pasta, I eat some sugars, I break those down, I get the energy out of those molecules. Right? And my cells use that energy to convert into ATP, and I use my ATP to do work. Okay? So I have my storage carbohydrates. Okay. And now these storage carbohydrates can be very, very, very long chains, but they're single or very slightly branched chains. Okay. And our body can break these down and open them up rather readily without much effort and harness that energy and use that energy as we need it. Okay. Now, other organisms, right, such as plants and bugs, rely very heavily on carbohydrates for structural components of themselves, right? And so some carbohydrates are very, very, very stiff and rigid and well formed for protective purposes. And so plants in particular have a carbohydrate structure called cellulose. And cellulose is what makes plants very stiff and rigid. Right? And the more stiff and rigid it is, the more cellulose it contains. And so if you think things like pepper skins and corn and heavy salads, okay, all those things have a lot of cellulose. They're hard to break down. We don't break them down very easily. The major difference between the storage polysaccharides and the structural polysaccharides comes down to a very, very little detail in the monomers of these sugars, right? And so when you're creating a molecule, you can have the same molecule with slightly different formats. And so it'll have the same chemical formula. Right? It's all the same components. It does most of the same thing, but there's very slight tweaks, right? It's kind of like being right-handed versus left-handed. And so when I look at this monomer of glucose, this alcohol group on this point here might be orientated up or it can be orientated down. Okay. And it's the same chemical, right? It's depending on the way that the branching started. But this very small, slight detail plays a huge role in whether or not I'm forming a structural polysaccharide or a storage polysaccharide. So in the storage polysaccharide, they're using all the same format, whether it be the up or the down version. Right? And so let's just say it's the down version. Right? And so all of my OHs are down. Okay? So that's for my storage polysaccharide. For my structural polysaccharide, they use alternating ups and downs. So they use different forms in an alternating pattern. And what this allows for is that these polysaccharides, which are coming off of branches, can now form hydrogen bonds between their groups. So there's an up and there's a down. Up and a down, right? And so they're near each other, they can form hydrogen bonds. And so instead of just being one single polymer strand out on its lonesome, these can branch into very large, very organized structures. And these large organized structures are much harder to break down. They're nice and solidly connected, right? And so they give that rigid quality of cellulose. This is also how bugs form their shells. They're chitin shells. They're made of carbohydrate structures similar to this. But this all comes down to this very slight difference of ups and downs in this format. Right? Same molecule, just slightly different position in this OH group. And so in the structural carbohydrates, the OH groups are alternating up, down, up, up, down, up. And that way, the ups and the downs can connect and form hydrogen bonds. Over here, they don't have that option. Right? At the very best, you could form like a 2-row setup. So carbohydrates, structural, storage, stores energy, forms structures. So now we're going to take a peek at lipids. And so lipids are things like fats and oils and waxes. And the monomer components of lipids are fatty acids. Now the way that we build a lipid, or rather a fat, is by taking these fatty acids, which are long hydrocarbon chains, carbon, 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 hydrogen, 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 hydrogen. Right? With these particular groups, I have an oxygen double bonded to this N carbon, and an alcohol group single bonded to this N carbon. Over here, I have a molecule called glycerol. If you notice, there are 3 carbons here, each with an alcohol group on the same side of the carbon. And so I perform a dehydration synthesis in this region here. I pop off of water, and now my fatty acid gets stuck to my glycerol. Okay. Now I have 3 regions here, therefore I can hold 3 fatty acids here. And now the major role of lipids in living organisms is as a long-term energy storage mechanism. So each of these peaks represents a bond between carbons, and between each and every one of those bonds, there is a reserve of energy. Right? And so by breaking down these large molecules into smaller and smaller units, we release energy that we use for work. And so these fats can hold a lot of bonds in a relatively compact area. Right? They're not as easy to break down as carbohydrates, so we can't use them directly as an energy source. We need to convert them into smaller units before we can break them down further into things that we can use to make ATP. But they serve a great, great function of storing energy for a long time. And so I can have fat deposits in my belly, right, on my cheekies. And those fat deposits are an energy reserve for the time when they're needed. And so this is how we build those fats. In a second, I'm going to return with some new diagrams, and they're going to take a little bit closer look at the different structures of different types of fats. Be right back. So, lucky you, I'm back. And on the board, I have 2 different types of fats. Okay. Now, they are very, very, very similar in structure. You can see I filled out all these C's now, so this is what our fat is looking like. I have my glycerol unit, I have my fatty acid units. Same thing's going on over here, glycerol unit, fatty acid units. Now the only difference between these 2 types of fats lies in the bonding of one of the particular regions of the fatty acids. So on this side, I have a saturated fat. Okay? And in saturated fats, all of the bonds within the fatty acids are single bonds. It's called saturated because it has as many hydrogens as possible. Okay? It's full of hydrogens. In the unsaturated fat, okay, I have a double bond in one of my regions. And so in this region, I am lacking a hydrogen. So I'm not full of hydrogens, so it's unsaturated. Okay. Now, it's a very small structural difference, but it's very, very significant in the way in which these fats move throughout our body. And so saturated fats, okay, because they are all uniform, have a tendency to stack on top of one another. Stacking. So like saturated fat can slide into another saturated fat and just link in, and they all stack. Because the unsaturated fat has this kink in it, they can't line up as flat and as nice, they're kind of janky, right? And so they don't stack up nice and flat. Yeah. So inside of our body, right, if you imagine these things floating around in our veins and arteries, because that's what they are doing, right, the saturated fats can accumulate on the floors and walls of our veins and build up and reduce and reduce and reduce and reduce and reduce, right, the volume of blood that can flow through. And that increases the blood pressure, leads to a heart attack eventually. Right? And so eating too many saturated fats is no good. Unsaturated fats are healthier. Right? And so when we see these things outside the body, okay, saturated fats are looking more like butter, okay, or meat fat. And unsaturated fats are looking more like oils, like olive oil or avocado oil. Right? And again, that's because of this. Right? So the form, that liquidy form of oils versus that more solid form of butter, comes down to the form of their small components. So because the fats can't stack up on themselves in the unsaturated fats, they don't form solids like butter. They are liquidy, like oils. These can all stack up, and so they form solids like butter. And they do the same thing inside of our body. Okay? So, a little bit more detail about the structure of the unsaturated fats. Now, as I said, typically speaking, natural unsaturated fats are much better for you than our saturated fats. Right? They have a tendency not to build up in our arteries and give us heart attacks and such. However, there is a form of unsaturated fat that typically does not occur naturally. It occurs through our processing mechanisms that we use to create certain products, such as shortening, right, where we take a typically unsaturated substance that is a liquid, like an oil, and we add hydrogens to it, and we change the structure of it to make it a solid. So you make shortening. These are things that are used in like baked goods and stuff. When you go through that chemical processing, what happens is you get what are called trans fats. And they're called trans fats because they possess trans double bonds. So the double bond that I showed you in the first image, right, that was over here before, was a cis double bond. Now these cis double bonds prevent the lipids from stacking up. The trans double bonds, however, promote the lipids stacking up. Okay? So this kink, instead of preventing the stacking in your arteries, actually increases the stacking in your arteries. Okay? And so this slight difference in chemical format, right, same chemistry, same stuff, just different format, different shape, right, leads to a vastly different outcome. Right? Tiny, tiny, tiny little difference, huge difference in function. Okay? So the trans unsaturated fats have a trans double bond. Okay? And a trans double bond is when the carbon peaks on the opposite sides of the double bond go in opposite directions. Now the typical natural unsaturated fat has a cis double bond. That's where the peaks on either side of the double bond go in the same direction. Okay? So cis unsaturated fats, good for you. Olive oil, avocado oil. Trans unsaturated fats, terrible for you. Okay? Fortunately, in this country, the FDA was like, yo, trans fats are terrible for you. Let's get them out of the food. So now we shouldn't have trans fats in our diets anymore. So, one last lipid group to talk about, and now this group is vastly different from the fats. Lipids in general are basically characterized as lipids based on their hydrophobic nature. And so structurally, this has very little in common with the fats that we just talked about, but it is very insoluble in water. And so that's why it's also a lipid. Now this group is steroids, and steroids are characterized by these 4 rings. Right? This is the base of the steroid. Now these R groups, right, which is a representation utilized to demonstrate where functional groups are attached to molecules. Right? They're variable, and depending on what R groups you have in these places, you get different steroids. Now, steroids play a very large role in our hormonal creation, right, in the way that our hormones function in our body. It's very important that they are not soluble in water. It makes them last for a long time in our system. It makes it easier for them to pass through our membranes. And so they serve very well as a chemical communication device. Right? And the fact that they just require these little modifications means that I can create the base, right? Lots and lots of the base, and then as I need specific types of hormones, I just modify the functional groups to create different hormones, right? So that increases the efficiency and the speed at which we can create these needed molecules. Okay? So steroids, just these 4 rings with different functional groups. On to our next macromolecule, which is proteins. So the monomer of proteins are the amino acids. And the amino acids have 3 subgroups. So I have the amino group, which has a nitrogen, 2 hydrogens. I have the functional group with this variable functional group here. Okay? And so this area is what determines the qualities of any specific amino acid. So if I have a polar group here, this amino acid will function with a polar nature. Right? If I have an acidic quality here, or acidic group here, this amino acid will be acidic in nature. So this region changes depending on the amino acid and gives the amino acid its qualities. And then I have a carboxyl group, right, which is a COOH. And then I have another amino acid here, same setup. So this is the general format of all amino acids, right? And now in living things, we utilize 20 different amino acids, and so we have 20 different functional groups that can attach here. Now, proteins are long chains of these amino acids, okay? And they form through a dehydration synthesis bond down here. Okay? So this region will be enzymatically connected. The water will pop off, okay? And then I form a bond between these guys. Okay? And bad place for that. Better place for that. Okay? So that is how I form proteins. Okay? I link together amino acid chains, very, very long amino acid chains. And basically the front group and the back group just serve as places to link. And this middle functional group is determining all the qualities of the amino acids, and therefore, in the overall structure of the protein's structure itself and its functions. Right? So these functional groups determine not only the function of the amino acid, but also of the protein overall. So just real quick, I wanted to show you some pictures, models that represent the shape of completed proteins. And so the structure of proteins are called the protein's folding, and there's different degrees of folding. And when we get to the transcription and translation unit of this course, we are going to look in depth at how the shapes of the proteins are formed and how they impact the function of the protein. But for now, I just want you to see that these are what proteins look like. Okay? So, the last group of macromolecules that we need to discuss are the nucleic acids, the monomer of which are the nucleotides. So here I have the generic format of a nucleotide. We have the phosphate group, which is bound to the sugar group, which is bound to the nitrogenous base. And now the nitrogenous base is varied depending upon which nucleotide we are speaking about, but this is the general format. Now, the two nucleic acids that should come immediately to mind are RNA and DNA. So DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid, right? And that's based on this sugar, right? So this is a ribose sugar. And DNA, the deoxygenated version of this ribose sugar, has a hydrogen at this second carbon, whereas the RNA has an OH at this second carbon. Right? And so it's a very slight difference between DNA and RNA structurally. However, as you should be aware, this slight difference makes it so that most RNA is single-stranded and most DNA is double-stranded. Right? And so this slight tweak in the general formula has that overall outcome for the functions of DNA versus RNA. Okay. Another slight detail that you should pay attention to is the numbering of the carbons. Right? You may remember the strand sense from Bio 1, that is to say that DNA is marked from the 3' to 5' end, or the 5' to 3' end. Right? And those numbers come from the position of the carbon. Right? And so this is the 5' end of the carbon, whereas this is the 3' end of the carbon. Right? And so it's kind of like the top and the bottom and the top. Okay? So that's where our strand sense notation comes from, is the numbering of these carbons. Okay? So that's our general nucleotide formula.
Excuse the interruption, Claire Stafford, please call 3449, thank you.
Bet you guys haven't heard an announcement in a while. You're welcome. So, a nitrogenous base can be any of the nitrogenous bases over here. And so on the left-hand column here, I have our pyrimidines, which are single ring units. So I have cytosine and thymine, which are found in DNA. And then I have uracil, which is found only in RNA. So cytosines in both, uracil is RNA, thymine is DNA. Now what I'd like you to look at is just how similar uracil and thymine are. The only difference is that thymine has a CH3, where the uracil has just the H groups. But otherwise they have the same functional group on this side, which is important to note because thymine and uracil are kind of interchanged with one another when you're moving from DNA to RNA. Okay, that makes sense because they have similar structures, so they can fit in kind of the same place. Okay. Now, over on this side, I have the purines, which are two-ring structures, right? And so we have adenine up top and guanine down the bottom. Again, same overall structure, just different functional groups stuck on in different places. Okay. You do not need to know the molecular structures of the individual nitrogenous bases, right? That's beyond the scope of the course. But what I want you to see is how similar molecules have similar structures, they fit into similar places. Right? It's also important to note that a purine always pairs with a pyrimidine down the entirety of our DNA, right? So a purine always pairs with a pyrimidine, which is important for the structural stability of the DNA molecule, because then you always have a 3-ring structure in equal distance across, so that we don't have wonky areas that are a little bit too long, a little bit too short, right? This uniform distance all the way down our DNA molecule helps keep it stable. Okay, I think that's it for nucleic acids. I will be right back with a model of ATP. Not going to talk much about it, just want you to see it. So, just a quick look at ATP, which is a nucleic acid. It's different than DNA and RNA in that it just exists as a single nucleotide, right? So it's only this one nucleotide with a slight variation in the way that its phosphate groups are attached. So rather than having a single phosphate group, like our typical nucleotides, ATP actually has 3 phosphate groups, hence the T, adenosine triphosphate. And this, of course, serves as an energy storing molecule. The energy that we put in and take out to do cellular work is stored in this last bond between the second and third phosphates. Right? And so this is actually a very high energy bond. Right? It's not the most comfortable molecule. Right? It's ready to break off. And so when we manipulate the ATP to release that energy, okay, so we release the energy and we bounce our ATP back down to ADP, which just has 2 phosphate groups instead of 3. And so when we go through the mechanisms that generate ATP, what we are doing is taking some free energy from somewhere, investing it, and we take a phosphate group, we shove it onto ADP, and we're back to having ATP. And so that's how this nucleic acid, this nucleotide, functions as an energy carrying molecule. And so that wraps up my 2-part lecture on macromolecules. It's important to remember that this overarching story is about how we get resources from the environment and how we get energy from the environment and how we use those resources to build our body, how we use that energy to maintain the organization within our body. Right? And so we have these macromolecules, which are made up of little uniform components called monomers. You link them together, you get polymers. You can break polymers back down into monomers. Right? And the chemical processes that do this are dehydration synthesis to build up polymers and hydrolysis to break down polymers. Right? And if we look at the smaller units of each piece, of each polymer, of each macromolecule, and we look at the shape, we will see that the shape impacts, or rather imbues, the overall molecule, the overall structure with its function. And so that's the big theme in this unit. Right? One of the big themes that we'll see throughout the course is that form determines function. Right? And we see that across all scales on any physical plane. Right? So as long as we're talking about physical things, the shape of something controls what it does. Okay? And so I want you to think about that when you look at these itty-bitty little differences. Right? Focus on those aspects of this unit rather than just the memorization of what things do. Right? What are they made of? That's not interesting. Right? Think about how what they're made up of is related to what they do. Right? Understand the cause and effect relationship. Far more valuable a tool set than just memorizing things. Memorizing things is a waste of time. Ugh, yuck. Learn stuff. Understand stuff. That's a useful skill set for anything. Okay? So, I hope you've enjoyed the second part of this lecture. I hope you've enjoyed the front of my classroom. Have yourself a fantastic day. Bye.
Talking away, I don't know what to say. I'm saying it anyway. Today is another day to finally shine. I'm coming for you, I love you. Take on me.